Welcome to a screencast introducing IRIDA, the long-term storage system implemented at QIB by the core Bioinformatics team. Andrea Telatine will now show you how to find your way around it.
Welcome to today's video tutorial on IRIDA, a powerful web-based application for managing sequencing runs and their associated metadata. In this tutorial, we'll guide you through the essential features of IRIDA, from logging in and viewing projects to adding metadata, creating new projects, and downloading reads. Let's get started. Before we delve into the specifics, let's take a moment to discuss our remarkable core bioinformatics team. Our core bioinformatics team plays a pivotal role in managing the QIB bioinformatics infrastructure, which comprises not only the IRIDA platform but also a comprehensive Galaxy instance for genomics analyses. Additionally, we offer a cloud environment that allows us to create customized virtual machines tailored to the needs of QIB researchers. Our core bioinformatics team provides regular training opportunities, equipping you with the skills and knowledge needed to navigate and utilize our bioinformatics infrastructure effectively. Finally, our team is here to assist you with your bioinformatics analyses. Whether you need guidance on experimental design, data interpretation, or troubleshooting, we are dedicated to providing the expert support you require to ensure the success of your projects. If you find yourself in need of assistance, we encourage you to utilize our convenient ticketing system. By accessing the web page displayed on the slide, you will find the option to raise a ticket by clicking on I Need Help or other links located at the bottom. The upper section of the portal, distinguished by its blue background, provides access to our KnowledgeBase system, known as Wiki or Confluence. Within this KnowledgeBase system, you can easily search for specific information. For instance, by entering the term training into the search bar, you will uncover a dedicated page featuring upcoming training events. So what is IRIDA? At its core, IRIDA is a comprehensive web-based platform that enables the long-term storage of sequencing reads in a well-organized repository. It allows you to securely store and manage your sequencing data, ensuring it remains accessible and easily retrievable whenever you need it. With IRIDA, you can store your reads, associated metadata, and even perform various analyses within the platform. It is designed and implemented to provide a safe space with highly redundant storage, compliant with the requirements for data storage of our funding agencies. All datasets produced by our sequencing core are automatically uploaded in the IRIDA server, and you will get access to the relevant projects. If you outsource the sequencing, you have to upload the reads to IRIDA by yourself or asking core bioinformatics to do it for you. Yes, you can raise a ticket for this. IRIDA offers all the advantages of a managed platform, and it's a system with both an easy-to-use web interface for easy access by all QIB researchers. At the same time, with command line tools like IRIDA Uploader and IRIDA Linker, you can automate the upload and retrieval of datasets from the command line. IRIDA is essentially like a local reads archive like EBI, ENA, or NCBI SRA, and like those it works better with annotated metadata. Adding metadata to a project is very easy, and this tutorial will cover how to do it. Let's now have a look at the interface. It's important to remember that this service is not using your usual NBI login, but rather a custom username and password. When Core Bioinformatics adds you to IRIDA, you will get an email with your username and a link to set your password. At the top, you will find the Projects menu to browse or to create empty projects. Clicking on My Projects, you will see a list of projects you have been given permission to see or manage. The system has a hierarchical structure. A project is a container for samples, and each sample can contain one or more sequencing files. We try to simplify this structure, associating to each sample only the output of a sequencing run. So a sample can usually have one file for nanopore or single-end Illumina sequencing, or two files for paired-end Illumina sequencing. Occasionally you might have more, but ensure this is expected. Here we opened a project named Galaxy, containing only 4 samples. Let's have a quick overview of the main tabs. Clicking on one of the samples would open its detailed view. The line list is a tabular view, which allows to access the metadata of the samples, downloading it as Excel file, or to add new metadata columns. We will see how to do it later. The Details pane allows to view or edit the information for the whole project. It's a good idea to add a meaningful description. Finally, from the Settings pane, you can add or remove collaborators from the Members section. Now let's explore the step-by-step process of adding metadata to a project. Metadata encompasses various attributes that we can assign to each sample, such as treatment, collection date, amount of extracted DNA, and more. As we said, after you open a project, you have access to the Line List tab to view and update metadata. Let's click on it. First, we export the metadata as Excel file. Even if the project is new, it already has some automatically generated fields like creation date of each sample. We can now open the Excel file. To avoid making redundant column, let's remove all the columns, keeping only sample and sample ID. Keeping sample only would suffice, but in this example we kept both. Now add as many columns as needed. Remember to double check what you prepare, as copy and paste errors are quite common. When you are done, save the file. Now, from the same page you downloaded the empty metadata file from, click on Upload Metadata. You can now drag the newly created file onto the rectangle, or click there to open a file selection dialog. A new page will list the column names extracted from the uploaded file. We need to select the key that will be used to merge the metadata, which is the sample column. When done, click on Preview Metadata to be uploaded to check if the process worked. Check the number of matching samples and the rows with unmatching samples. If everything looks fine, click Save Valid Metadata, and you will see how the new information has been added to the database, and it's now visible from the Line List tab. Finally, let's briefly introduce some advanced topics. Here the goal is to give an idea of the process and pointing you to check the documentation from the service portal rather than guiding you step by step. The first topic is how to upload reads to IRIDA. If you have just one sample or two, you can do it from the web portal, but the recommended way is to use the HPC where the software to do so is already installed. Before using it, you will need to create a configuration file with your authentication details, as described in the documentation. First, we need to create a new project, as we will need to have the correct project ID before uploading the reads. Let's give it a name and fill in the appropriate description and information. When it's done, take note of its numeric ID. Now we can open our terminal, log in the HPC, and then navigate to the directory containing the reads. We must start an interactive session or IRIDA uploader won't work. Source the package secfu and IRIDA uploader. We will use secfu to automate the generation of the sample list. With secfu metadata, we can create a file that we must call samplelist.csv and save it in the directory containing the reads. This is how it looks. Finally, with uploadrun.py, we can pass the directory with the reads and the metadata, and the program will start creating the samples and then will upload the reads in each sample. The procedure may require some time to complete. Once finished, you should observe all the samples and their corresponding reads. It is essential to verify that the samples have been successfully created and contain the expected reads as intended. Once the reads have been archived, they can be used in different ways. You can simply download them from the web interface. Again, this is a viable procedure only when the sample number is very limited. Alternatively, you can download the reads to the HPC using a package called IRIDA Linker 1 0.0. Similarly to IRIDA Uploader, there is the documentation portal with instruction for the setup and the actual download procedure. Finally, if you use Galaxy, you can simply import reads from an IRIDA project to Galaxy itself, as you will see in the dedicated tutorial. Remember to check our documentation, and if needed, to raise a support request. We can help you configuring your client or simply uploading your reads to IRIDA.
You deserve your cup of coffee now. Well done for watching the video. We hope this video tutorial is helpful. Now check the description for links to documentation and support portals. Bye.